Hello everyone, I am DR.R.UMA Maheshwari, Assistant Professor, Department of Biotechnology, Cauvery College for Women, Autonomous. Going to explain about the cell. The cell is the structural and functional unit of life. All living organisms are of cell. Cells are of different shapes and different size. There are many cells in our body itself. The muscle cells are striated in nature. Intestinal cells, which is having mucus, hair, and liver cells, it is amoeboid like structure. Cardiac cells are filamentous in nature. In our body, the longest cell is the nerve cells, which is having dendrites, and the blood cells is biconcave in structure. Based on the cellular organization, cells are classified into two types. One will be prokaryotic cell and other will be eukaryotic cell. For the prokaryotic cell, bacteria is the example. And the case of eukaryotic cells, all the higher organisms like plant, fungi, plants, animals, they will be coming under the eukaryotic cell. See the structure of prokaryotes and eukaryotes. Let us see about the cell discovery. Robert Hooke is the first person who discovered the cell from the plant Quercus. He discovered the dead cell. That is, he isolated from the bark of the tree Quercus. Next to Robert Hooke, Anton von Leeuwenhoek discovered the living cell. He discovered the microscope as well as the bacteria at the first time. Then Robert Brown discovered the nucleus. About cell theory. Cell theory was proposed by Schleiden and Schwann. Schleiden worked on plants and he found that all plant cells are made up of cell. And Schwann, his co-worker, he found that all animals are made up of cell. So they both together proposed the cell theory that all organisms are made up of cell. Cell is the structural and functional unit of life. Next to Schleiden and Schwann, modern theory was proposed by Rudolf Virchow. Rudolf Virchow said that all new cells are arising from pre-existing cells. He said that omni cellulae e cellulae. In the prokaryotic cell, bacteria is the example. Let us see the structure of the bacteria. Bacteria has cell wall and also outer covering, which is called as glycocalyx, which gives protection to the bacteria. It is a mucilaginous substance. and the nucleus will be primitive in nature, so that we are calling it as a prokaryotes. There will be no histone proteins and there will be no the structure of chromatin, only the chromosomes scattered over the cytoplasm, so that the prokaryotic nucleus are called as nucleoid. This is the structure of typical Another specialized structure is mesosomes. It is the inclusion of plasma membrane which does all the metabolic activities, and this mesosomes is not present in the eukaryotic cell. Another unique structure is the plasmid. Plasmid is the extra chromosomal self-replicating double-stranded DNA which is present only in the prokaryotes. Nowadays, in the cloning purposes and genetic engineering purposes, we are taking this plasmid as a tool. In the prokaryotic cell, bacteria is the example. Let us see the structure of the bacteria. Forms of bacteria. Bacteria in the structure also they get varied, and also outer covering which is called as the glycocalyx, then which gives protection to the bacillus is a rod-shaped bacteria. It is a mucilaginous vibrio is the gamma-shaped bacteria, and the nucleus will be primitive in nature, so they are calling it as a prokaryotes. And there are different types of cells. There will be no histone prokaryotic cell, and there will be no the structure of chromatin, cell, animal only cell, the chromosomes scattered over the cytoplasm, so that the prokaryotic nucleus are called as nucleoid. Another specialized structure is mesosomes. Thank you all. It is the inclusion of plasma membrane. We have seen the structure of prokaryotic all cells, the metabolic activities. In the coming part, and we will see about the eukaryotic cell in the eukaryotic cell. Thank you all. Another unique structure is the plasmid. Plasmid is the extra chromosomal self-replicating double-stranded DNA which is present only in the prokaryotes. Nowadays, in the cloning purposes and genetic engineering purposes, we are taking this plasmid as a